Biosensors. Biosensors we use in everyday life. We need biosensors for so many things. We start from uh, our food, contamination in the food, we need biosensors there. For homeland security, for protecting ourselves, our populations, we need biosensors. For research, looking at levels of different things and how do they affect us, we need biosensors. There's some examples given, but of course, today COVID-19 has made it clear for all humanity we need biosensors. We need good biosensors. We need nano-biosensors. We need sensors which are selective so they don't give false results. Sensors which are sensitive so they can detect very, very small amount of the target entities that we want. We need biosensors in our clinics. We want to test what's happening between in our body, what's maybe to detect a disease, maybe to detect the result of therapy, how our body is reacting, maybe to detect new drugs, new vaccines. Now we know vaccines, why they're important. We want biosensors to detect environmental things, maybe water, maybe air quality, maybe exposure to uh, bacteria, exposure to chemicals. We put things in our ground, fertilizers. So we want to know, we want to sense how things are happening. So all across the board, we need biosensor. So a biosensor is basically a device that detects and selectively detects. So that's where the recognition element comes in and reports the presence or the exact quantity of a biological agent. And this detect implies recognition, selectivity. Another very important aspect is sensitivity. We'll look at these concepts today as well. So we do recognition by many means. Maybe it's a through a molecule, maybe bunch of molecules, maybe a cell, maybe change in some enzymatic activity. But for all different kind of things, we have different kind of recognition elements that we may use, right? So we may have a bacterium that we're looking, maybe we need to coat antibodies on a surface, maybe we coat some proteins. So whatever we do, there are some basic components of that we want to target, we want to transduce, convert it into a specific signal so the end user can look at the signal and make a judgment, right? So give me an example of a classic biosensor that we all have, and it has these qualities. It helps us avoid bad food from its odor. It recognizes people if somebody is around the corner. It can even detect uh, things that may be poisonous or indigestible. It can tell you even if there is excessive heat or cold, helps us survive.
Like a thermometer.
A biosensor that we all have.
Hmm.
How do we detect unpleasant odor?
Oh, the smoke alarm and like your smoke alarms in your house. No.
Julio,
Yeah.
look at me.
Yeah. Okay.
Nose.
A pen?
Nose.
The
Think
pen?
about nose,
Oh. Okay. Yeah, I get it. I get it. I got it.
think about tongue,
Yeah. 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 Yeah.
right?
Yeah.
So think about eyes. Our eyes burn if
Yeah.
you're
Yeah.
exposed to
Uh,
some gas, right?
yeah. I was
So,
thinking more like mechanically. My bad.
right, right. But you're
Yeah.
right. We That's what we call biomimic or biomimicking or biomimetic sensors, where we want to emulate
Yeah.
what living things may be doing already. So here's an example of olfaction, right? Just breathing, just uh, looking at maybe so analyte. There's orders that we want to detect in it. So how it does? We have receptors, neurons, and that's an actual picture of olfaction nerves. How does it do? So we have liquid at the interface. So here's mucosa. It says cilia and mucosa. There's a liquid, and the gas comes in contact with that liquid. The liquid takes the molecules of that air, dissolve them, and then there are neurons which would fire the data to let the brain know if the thing is okay or not. This is a classical example. Now think about it. How do we replicate that? Would it be easy to replicate this? Essentially, if we can create a bio nano interface here and we create a detection system, we can replace it. It's not easy, but this is something where a lot of people have been working. If you just look up e-nose, you will find out many eh, people and lots of funding that have gone into this. That how can you uh, emulate what the nose of a dog does, for example, right? So then if you can come up with something, we don't have to send living things to detect those. Maybe it's a explosive, maybe it's a virus, maybe it's some toxic material. You can send that device on a robot to detect those things. So, but it's a classic example of a biosensor. Recognition, we have discussed this before, it can be done by many different ways. So today we'll look at some devices and some recognition elements that have been used and shown to do sensing at uh, molecular scale. Antibodies are what all our bodies make. So those antibodies, they bind, and basically that's the reaction to something that may be invading our body, right? So they recognize antigen and they inactivate the antigens. They are generally Y-shaped molecules, and they have what we call two identical long polypeptides. So polypeptide is repetitive chain of molecules. They have two long and two identical, which gives them that Y shape. And these are three-dimensional structures which are built up of uh, smaller molecules, amino acids, and they bind to receptors on specific cell surfaces. So they would the cells would have receptor molecules and they would bind to them. And there are many different antibodies that are known, right? There's IgG, IgA. So we use many of them to make our devices selective to specific molecules that we are looking for. And there's lots of research that has gone in into modeling these, into finding exact location of which molecular part is at what space, at what place in there. And that information is what we would use then to engineer a device surface or engineer the sensing capability according to the, the structure, the folding, and by finding out their specific binding capabilities. So this cartoon is showing that you have the antigen and you have the antibody that comes and bind to it and makes a three-dimensional structure by those uh, chain binding. And those interactions can be quantified. We have specific affinity between two different molecules that we defined in numbers. So, and there are formulas how we do that. Don't want to go into details, but for any two molecules, there is a strength of how strongly they would bind. And there is specific numbers that we use to explain the binding affinity. So if they are just randomly binding, so there would be the there'd be binding and unbinding. So the numbers would be uh, same for them. But if they bind specifically, we have a number which is very huge number per mole. Mole is Avogadro's number, so we find exactly how many moles are there. So that affinity constant tells us if it's big number, the affinity between those two molecules is higher, which means they would strongly bind to each other. So it's a way of looking at of identifying which molecule would be good if you want to detect something, may say called X. The Y molecule would be, if it has high affinity, you would use the Y molecule to bind it, right? So there is a whole uh, research behind, or there are numbers associated with these reactions that we use to design our biosensor. And if you if you break it down at the very bottom, these are simple basic bonds that give that affinity. There are a number of different bonds we know the hydrogen bonds, ionic bonds. So this picture on the left side is breaking down some of the kind of binding that happens. There may be hydrophobic interactions where they want to repel water, so they come together. There may be van der Waals forces which bring two molecules together. So, but when all these things come together, four types of bond, you get an overall affinity between two big molecules. So here, the antigen, the receptor on the surface would bind to the antigen binding site of the of the protein, and that binding would be composed of these covalent forces. So we take advantage of this affinity and we coat our devices. Some devices we can readily understand, some devices may be a little bit complex in their design. So here's one device, cantilevers. Cantilevers is a very small micro scale jumping board. You know, on swimming pool, you have this diving board, which is bound from one side and it's free from the other side. Cantilever is a very interesting device. Mechanical engineers study it a lot. And the idea is that for this kind of structure, there are forces which may be acting on the where it is bound with the wall, and we like to calculate those forces. But in this kind of application, what we are doing is, since it's a very small element and it's suspended in the air, you can use a bunch of things. You can measure its bending when it bounds to some molecules, like it's shown here from going from figure A to figure B. The red molecule binds specifically to the green target. And when it binds, you see that there is weight change and it shows bending. The blue molecule, on the other hand, binds to some other molecule shown as a yellow molecule here, right? So what we are talking about is maybe as simple as cholesterol, right? It's a molecule, right? We want to find out exactly how much it may be in a unit volume. Or it may be as exquisite as a brain tumor biomarker. We want to identify if a person has brain tumor, we take a solution and we want to run it, and we want to look for that biomarker or that molecule which is known to be associated with some cancer. So that's one example. We would see bending, right? Now, bending can be detected by many ways. Maybe we are looking at the uh, resonance frequency, and when the weight changes, the resonant frequency would change. That's one phenomenon, resonant frequency. We know everything in nature resonates at some specific natural frequency. That natural frequency can change if the property of that material would change. Or we may just point a laser to that and we see the reflection of the laser like atomic force microscope. And if it bends, the angle of the reflected laser would change, right? That can tell us the if something has bound there or not. So here, if you see on the right hand side, the upper one is showing there's been uh, two, three molecules have been functionalized on the surface, and then there are free chains. Once they bind, they are shown cartoon on C and D that that binding would result into change in the mass. The lower picture shows that when they introduce a molecule, one uh, cantilever and the other, they are showing that differential signal between those two. It's given in nanometers. So whatever we use, basically we would be looking at change in signal because of binding. Here there are more examples of using cantilevers and looking at the stress, right? Again, because of binding. So PSA is prostate specific antigen. Men over 40-year-old are recommended to get their PSA levels tested because that tells us if a person has the maybe has prostate cancer, right? So there's debate. Some people may have genetically higher level of PSA but no prostate cancer. Some people may have genetically, they have cancer and they may not have PSA. But overall, it's a first line of defense. We get checked for PSA. Now, if there is very small change in PSA concentration, we need this kind of sensor. So if you see on the left-hand side plot, it shows PSA concentration and then y-axis is showing the deflection nanometer. Again, introducing small amount and then seeing the change in the properties of cantilever, right? So bunch of examples of how we can use these sensors where we functionalize them and we look at this. This is an example where they are showing differential biosensor. So basically they have these interdigitated arms here shown. It's not shown in this picture, but that's, if you look at those papers, so this region is where they have those differential things, which means that one side would be bound. If it's bound, it would change the distance and you can measure the difference in deflection because of the binding unit. Any questions along the way? We are building up on top of what we have studied so far, right? So I hope looking at your uh, GRPs and uh, your review papers, I'm very confident you guys can understand what's going on in these examples. So again, here, now aptamer is a molecule that can bind selectively to some entities. So here, thrombin molecule is the target, blue, shown in blue in this plot as well. So thrombin is a biomarker again. Now, this is work from Purdue University. I think one of you talked about work from Purdue in your one of your assignments as well. So if it binds, it changes shape. If it doesn't bind, single-strand DNA stays as it is. There's work on E. coli detection as well. So they break apart E. coli, that's what's called lysate, and they look at the molecule, what's inside of that bacterium. And that concentration can be seen by, again, differential deflection in the cantilever, which shows that a spike. When you add buffer, we see spike. When you put the cell lysate, we see spike. When you put another Taq polymerase, we see spike. And then you wash it again, you see spike. So those changes in deflection would be indicative of the presence of that molecule. There's work done with the virus detection as well. So here, the surface is coated with specific antibodies, and when the antigens are captured, there is a change in the, they are looking at amplitude here in absolute units. So y-axis is the amplitude of the movement of cantilevers again. So this paper in PNS, I did some imaging for this paper. They have published my electron microscopes in this paper. So this work was done with vaccinia virus. Essentially, if we have antibodies for whatever virus, we can use it to detect those viruses using this kind of signal. So cantilevers have huge application, many places where they have used cantilevers. So here in this picture, this black area, this gray area is the top of the device, and this black is the hollow thing, kind of the swimming pool, if you can think. And then these fingerish things are the cantilevers which are suspended, and there's hollow place between them, which is shown in black. So once things land there, then you see the change in the signals, uh, like let me, like shown here. So here they are measuring frequency, and with the amplitude and frequency, they can see the frequency shift. So it's resonant frequency. When the virus is bound, they see change in the amplitude at different frequencies. There's work done between by measuring the electrical current between two electrodes. So what's happening here is that they have two electrodes, right? So if you short the electrodes, you would get Ohm's law, right? Voltage versus current. But you have made open electrodes, and then you put the molecules in between, selective molecule that are supposed to bind to the target molecule. So when the binding occurs, they introduce nanoparticles to increase that the conductivity. So once these come with selective binding, you get the increase in current between those two electrodes. So basically, ramp the voltage and measure the current. That gives you change in current, change in resistance. There is AC measurement also done, alternating current, where they ramp up the frequency like we saw in the previous slide, and you can see the change in frequency over a range. So whatever mechanism we'd be using, we get the signal out, transduction. So from the interface, which is the left-hand side, we have to now focus on the transduction. What kind of signal can we measure? There is a whole range of signals that we can measure. Luminescence, change in light. We can introduce a molecule, or we can have a molecule that undergoes changes and maybe gives out light. So we can measure change in light. A lot of time we do that. The reaction would be measured from the change in the production or emission of light. There may be we thing called surface plasmon resonance, which is an optical excitation of a surface, and that results in looking at the physical changes on the surface. So we plasmons are basically light waves that when you throw them on a surface, they get captured right next to the surface and they travel alongside the surface for a long period of time. Now, once it's traveling, and if the quality of the surface, the binding of the surface changes, there is a shift in frequency of plasmons. So that's another way how we can measure the wave on the boundary of the uh, sensor and the external medium, right? Change in the binding activity that can be measured with them. Now, there's whole bunch of structures and things that can be made at small scale to look at those small changes. We may not be able to look at all of those, but we can look at some of the engineering phenomena that are interesting for us, because that can help us understand or design biosensor at small scale, right? So we fabricate, and we have seen that. So we can nano fabricate biochips and biosensors. We can attach molecules. We have seen that, and we can recognize them. Now, what are the uh, conditions or situations that at that molecular scale that are interesting for us? Because they're different from larger scale. Fluid flow, for example, is different. We know fluid flow or through our tap, right? You open a tap, you get the water. We know fluid flow in capillaries, right? We prick the finger and we take the wick the blood and put in a glucose sensor. These are large scale fluid flows. But fluid flows at micro and nanoscale are different. And why is it important for us? Because for any sensor, we have to look at what is the, for example, like in red, it is shown a metal electrode. It may be the surface, the sensor surface. We want to functionalize and we want to measure the biomolecules from liquid, right? So now what happens at that nano-bio interface? That's of our interest. And here I'm showing one important property, which is called electron double layer. Electrode and electrolyte interface. The thing is that in sensor, for example, metal, electrons are free floating. They can easily move around. Now, in the solution, if you have overall positive charge or negative charge, whatever charge you have, if the charge accumulates on the surface, that's going to change the charge distribution in the metal. So, for example, here it's shown that water molecule, this is water basically, H2O, 2H plus and 1O. They, of course, they are in contact with the surface, and that causes the electrons to form close to the surface, which means there is a new added barrier to the interface. So your interface is now becoming thick because of reorientation of charges. If you look at, and it's a very uh, qualitative picture, the space charge can be written as this. So the charge is not evenly distributed. Unlike if we would have thought that the charge would have been same, no, we see that the space charge is, it takes some time before it gets to the background charge. So on the surface, vicinity of the surface itself, there is an accumulation of charge, positive or negative, it doesn't matter. But what's the take home message is that at nano scale, interface has its own issues. Now, this coupled, now if this is happening inside a small channel, fluid flow, we cannot take the flow as we take at larger scale. In larger scale, we say, okay, solid doesn't flow, fluid is that can, under small shear stress, it can start flowing, right? So solid needs more force, fluid requires less force. Now, when it take it to microscale, what do we do in microscale? We have MEMS devices where we have a motor going on. There is friction. We are pushing air to remove the heat. Maybe we are transmitting force from one place to the other, hydraulic. Maybe we are creating a jet propulsion. Maybe we are transporting molecules. So these are large scale things which I'm showing, but I'm talking the nanoscale equivalent of those things. When we go to nanoscale, we have, because there are different forces involved, we have to understand different phenomena at that scale. At micro scale, again, heat, we may be uh, moving things, we may have very small openings where we are pushing fluid through. For example, desk jet printers. We use laser printer, and then there are inkjet printers. So the inkjet printers, The the head has microscale holes in it through which the ink comes out. So that's a force that's coming out of those microscale holes. We might be transporting liquids, biological samples, for example. So when we go to nanoscale, here we're looking at microchannel and nanochannel. You see the surface charge becomes much more important, and which causes there is potential buildup. For example, between C and D, the green line is showing the amplitude of the potential. In C, with the distance, before you get to the other boundary, it goes to the background potential. But in nanochannel, the other boundary comes before it gets back to the background. And E and F is showing ionic concentration. So ions are accumulating close to the surface, but when you get further away, you get the background ion distribution. But that does not happen in case of nanochannels. A few physical phenomena that at large scale we treat these things as continuous, but when we talk about uh, nanoscale, they those things break down. For example, we take no slip conditions in many of our macro scale fluid flow. We never consider the surface friction, but when you go to nanoscale, we have the surface friction comes into it. So there is a quantity called Reynolds number, which is used to uh, identify the inertial forces. So if you in nanoscale devices, we call them low Reynolds number because their uh, inertia or the fluid flow has more resistance to the flow because of the small size. Going too fast, just few ideas on we know nano, we know bio, we know why we want to do it, but then we have to understand what are the issues at the interface of two things, right? Now, from continuum, when we go down, there is a thing called Brownian motion. Things move around freely in space before they encounter or uh, they strike another molecule. It's called mean free path. In larger systems, mean free path is the dominant force. We say okay, the molecules or atoms can travel certain distance before they strike another of those things, and they can change paths. But when you talk about nanoscale systems, now we are putting boundaries closer to their mean free path, which means the theory that we have for large scale breaks down. Now we have to average on over the smaller length scales. So now because of charge accumulation, we have current that starts flowing. So this thing called uh, we have pressure driven flow, but then there is another thing called electroosmotic flow, which happens to the phenomena called electroosmosis. Because of the charges, there will be a flow of, because of the surface attached charges or charge, which is the electron double layer that I was talking about, there'll be a molecule layer which will be moving in the other direction than the actual physical pressurized flow. And then if you add on top of that molecular recognition in those small scale devices, it becomes even more complex to understand or to model that system. So this is what we would call the molecular SAM and its effect. So going from large scale systems to nanoscale system, we have a totally different physics involved and totally different set of equations that come. Here's another example. We know filtration, right? Filter, you get your car filter changed every 3 months or so. right? We have filters in water system. We change the filter. What does a filter do? What is a
Takes out impurities.
it removes impurities, right? Basically, it removes particles, right? And the particles that it removes are bigger in size than the filter pores, right? It's a physical filtration. Now, here's the thing: if you make nanoscale membranes, this is what's called active membrane. These filters can filter out things based on their chemical nature instead of physical filtration. So they think about it that you have a solution and you put a filter and you expect the solution to go through and physically things that are larger to stay on this side and everything else can go on the other side. But if you coat a membrane, functionalize a membrane, it may stop even smaller molecules to go through and let only molecules go through that chemically bind to that membrane. So it's a device or a system where in bio application is that we may have a solution which have lots of different molecules, but we are interested in one type of molecule, red molecule, for example. Oh, sorry, target molecule triangle is shown here. Now I want to remove everything and get only the triangle one so my sensor can detect it. So that's something which this kind of device can do. And I'll just skip through. There are a few much more detail, but let me just share the results and show the idea of what we can accomplish with this thing. So we can accomplish that we introduce a mixture of bunch of things, but it filters, chemically filters only the things that are of our interest. For example, the green thing. Although you see the blue thing is also smaller in size, but it would not let the blue thing go to the other side. Only let the thing, and green thing can be a biomarker, any molecule which is important for detection of some disease maybe, right? So that's the another application of uh, nanoscale fluids where we can separate out things, filter out things, not based on the size, but based on the chemical nature. All right, and I'll give you another example and then we can wrap up. This thing called nanopores or ion channels. You have seen ion channels on nuclear pores, right? Where the molecules go in and out. We looked at those in cells. We have seen cell walls have some ion channels, holes through which the molecules go through. We can emulate them and make devices which have very small holes. We can make a small thin membrane and drill a very small hole. And with that, we can characterize DNA. We can characterize DNA molecule as it goes through. We can measure uh, electrical signal. We can measure DNA with DNA proteins, biomarker drugs, bunch of things. How it does is, so we put two electrodes on two sides, like for in this example, and we measure current, right? Ionic current. Ions are flowing, but they flow only through that ion channel, which is shown here, right in the middle. You can see my arrow, right? So now when a DNA molecule passes through, it blocks the flow of ions. Basically, that results into this reduction in current. So this reduction in current or this pulse or this negative spike is exactly because of these DNA molecules. So what we are doing is we are measuring passage of one DNA molecule. And think about it, DNA molecules 2 nanometer wide and few bases long in this case, as much as they have designed it. But with this device, we can measure exactly one molecule. Here's another example where it was not they, it was being many years ago. We did make a device which could measure single base mismatch. Think about it, that's the ultimate level of mutation, one base mismatch. Many bases mismatch is a bigger mutation. This was where we could detect, and you can see the difference in signal. You see, perfect complementarity shows many, many, many more spikes, which are much narrow. A mismatch shows fewer spikes and bigger spikes. And that data can we can use some equation to understand that data as well. So at the end of the day, again, you can model it and you can show that if a molecule is binding, it shows different kind of pulse. If it is not binding, it shows a different kind of pulse. So we take molecular recognition elements, port our devices, and gives us biosensors which can detect things at nanoscale. All right. So, and in all cases, you can measure the data and you can plot to give a output or a result for a person who can utilize these devices. All right.